So the title of my talk is how SARS-2 variants are selected and how they are spread, and how this relates to their spread during different phases of the current pandemic. Next slide, please. So most of this data is all background data is already known. So COVID-19 has been responsible for almost 1 billion infections and 7 million deaths as recently as a couple of months ago. The pandemic severity was actually controlled by a couple of important actions. One was stringent public health measures were introduced very early in the pandemic to restrict transmission. This involved severely restricting the movement of people, closure of schools and universities and all sorts of workplaces. Then also the rapid development and administration of vaccines, which was started late in 2020, together with innovations in clinical treatment to mitigate the severity of the symptoms in those who were hospitalized. And this included dexamethasone and therapeutic monoclonal antibodies administered to severely ill patients. Were it not for these measures, the number of fatalities would have been much, much greater. Now, SARS-CoV-2 variants have risen sequentially and spread worldwide to replace the original Wuhan 2019 strain, which was the one that caused the outbreak in December 2019. New variants keep arising even after 70% of the global population has received one dose at least of a COVID vaccine, and over 13 million doses have been given worldwide by the 8th of May 2023. Next slide, please. So next strain, which maintains an open repository, and you can access, everyone can access that in the link that's provided here at the bottom of the slide. The next strain maintains the sequences of all the known sequences from all the different parts of the world and analyzes them at regular intervals. So we know that the genome is the RNA genome, which is a single-stranded 30 kilobase genome, codes for about 29 different proteins, structural and non-structural proteins. And this diagram from next strain shows the location of the gene for the spike or S protein and the variation in amino acid sequences. And you can see that the variation in amino acid sequences is most prominent in the spike protein, which of course was the basis for the early vaccines, or many of the early vaccines. Next slide, please. So I won't go into detail. The spike protein is a very large protein, 1,273 amino acids long. It's a protein that has certain distinct features, two domains, S1 and S2. The S1 domain contains the receptor binding domain that binds to the ACE2, the angiotensin converting enzyme 2 receptor on host epithelial cells. It also contains an N-terminal domain, amino acid residues 13 to 304. And the spike protein also contains a membrane anchoring region. Now, host cell proteases, furin, cleave at the S1-S2 junction, and that occurs during formation of the virions. And a transmembrane protease, TMPRSS2, cleaves within within the S2 domain to activate membrane fusion. SARS-2 can also enter the epithelial cells through endocytosis rather than through binding to the ACE2 receptor. And this involves cleavage of the fusion of S protein by cathepsin and fusion of the virus and endosome membranes. Infection also spreads between cells by an S-dependent membrane fusion event. So that increases infection in the epithelium. Next slide, please. Now, the couple of features about the immunity. First of all, inhaled virions typically infect the nasal epithelium first, and then the rest of the upper respiratory tract. We know that innate and adaptive immunity, innate meaning non-antigen specific generalized immunity that all cells and tissues are able to produce. Adaptive immunity involves antigen-specific responses involving T cells and B cells. B cells produce antibodies. So we know that innate and adaptive immunity in the upper respiratory tract can eliminate the virus with little or no symptoms. If one does not eliminate the virus successfully in the upper respiratory tract, then the virus moves on to the lower respiratory tract and causes severe disease. We know that genetic defects in the innate immune receptors and signaling pathways, these are inherited defects, increases susceptibility to infection in those persons who suffer from such genetic defects. That's well established. And innate immune responses then kickstart the adaptive immune responses. We also know that population genetic differences, for example, in the nasal ability to warm air, which is essential for innate immunity, immunosenescence, the age-related loss of immune activity, and these immunosuppression also influence the immune response and infectivity of SARS-2 in the upper respiratory tract. Details of these have been described, and you can look up the relevant publications for that. Next slide, please. So what I want to focus on is how do mutations arise in the SARS-2 proteins, and how are the variant viruses selected, variants that carry these mutations. The mutation rate in SARS-2 is relatively low, and that's because there is a and as we saw earlier, it's particularly prominent in the spike protein. The mutation rate is low because the RNA-dependent RNA polymerase, which copies the genome, has proofreading activity that cuts away most of the mismatched nucleotides. However, this enzyme RDRP polymerase also facilitates recombination between RNA molecules if two different SARS-2 strains infect the same cell. So it can increase variability in that sense, in that way. Now, what then happens is that natural selection acts on the mutant viruses, and especially those mutant viruses that show increased infectivity, evasion of immune responses, and increased transmission, are able to replace previously present less competitive strains of SARS-2. Now, the transmission is rather complex. The transmission from one infected person to another is rather complex. It depends on the rate at which the virus infects the host cells, the rate at which it multiplies within the host cells, and as we will see later, how it's able to evade immune responses, innate and adaptive immunity, and then produce an increasing number of viruses that are in the host cells, which are then able to be transmitted. So they're very complex process, and many factors are involved. And we should focus only on some of the important ones. Next slide, please. The next strain has maintained the rate at which different variants arise, and we're talking about the major variants. So from early January 2020, a number of variants circulated in the world, and Alpha became very prominent in late 2020. It was replaced then by Delta, which reached almost nearly 100% infectivity in the world, infections in the world, and Delta was then replaced by Omicron, which has spawned a number of subvariants. And the Omicron 23A, we should see, is the one that is circulating, and 22E are the ones that have reached almost 100% prevalence throughout the world. Next slide, please. The phylogeny of these virus variants is rather complex. And next strain is able to demonstrate that in this figure. And you can see the early virus strain, which which arose in 2019 here, and the more recent ones are the Omicron XBB and Omicron BQ1 here. So, can you see my pointer? I hope so. Anyway, so there are a number of different nomenclatures. Firstly, the next strain, next clade nomenclature, which is based on a number, depending on the year of detection, and an alphabetical suffix, which gives the order of detection. Then we have the Greek terminal name, alphabetical names assigned to these variants by the World Health Organization. And finally, we have the Pango nomenclature, which has a alphabetical prefix and a number which shows the relationship between the different variant lineages. These are not perfect classifications, but they are indicative. So we can see, for example, that familiar variants like Alpha and Delta, which I'm going to talk about in detail later, Alpha, Delta, and Omicron in this lineage map, which is maintained by next strain. Next slide, please. Now, I'm going to divide the pandemic into different phases to discuss variants, and I'm going to take the most important variants that arose during each phase. First of all, the pre-vaccination phase. Vaccination was started in December 2020, and before that, there was a whole year for the virus to evolve. And one of the major features of the evolution of a variant carrying a mutation, a D614G. This was not given a name, but it arose simultaneously in many countries and spread soon after March 2020. So D stands for aspartic acid, amino acid, and G for glycine. So this was quite a drastic mutation, a negatively charged amino acid to a neutral small amino acid side chain. And this had the following feature. This mutation caused the RBD, the receptor binding domains, in the trimer of the spike protein to be in what's called an up conformation, a slightly different conformation, which permitted it to bind more readily to the ACE2 receptor on respiratory epithelial cells, leading to better membrane fusion and therefore increasing the infectivity of the virus to the respiratory epithelial cells. So this gave it what you may call a greater fitness. As I said, the fitness is rather complex, but increasing infectivity is one strong aspect of the fitness. And that was acted on by selection, presumably, and leading to its occurrence in a number of isolates. In mid-2020, the Alpha variant was identified in the UK. And this then spread rapidly to become the predominant variant in many countries. And this was associated with additional mutations, more than the D614G. And this included the N501Y, that's asparagine to tyrosine, and a deletion of histidine 69 and valine 70 in the spike protein. And these two mutations were shown by using artificial viruses to increase infectivity, transmissibility, and also, unfortunately, pathogenicity. It's certainly more pathogenic than the previous variants of SARS. And the increased transmission was the primary driver for selection at this time during the pre-vaccination phase. Next slide, please. I shall now move on to the early post-vaccination phase. And I'd like to use the Delta variant as an example here. The Delta variant emerged in April 2021 in India and then spread worldwide to become the dominant variant. So the Delta variant contained additional mutations in S compared with Alpha. In particular, mutation L452R, which was a leucine to arginine mutation. That increased the stability of the spike protein, the viral infectivity, viral fusogenicity with the host cell, and therefore viral replication and fitness. Additionally, there was a P681R mutation, proline to arginine, which increased the membrane protease-mediated cleavage, fusogenicity, and syncytium formation. That is the ability of respirators for the virus to spread between respiratory epithelial cells, causing the formation of a syncytium of cells, where the membranes break down. And that is associated with greater pathogenicity compared to Alpha. And there was a very much higher risk of severe COVID-19 with the Delta variant compared to Alpha. And in fact, in New Delhi, Delta breakthrough infections were observed during this period in people who had been vaccinated with Covaxin, which is the whole inactivated viral vaccine, and Covishield, which was the adenovirus-based spike protein vaccine. So clearly, these vaccines were the Delta variant was able to evade in some people the protection conferred by vaccination. Next slide, please. Finally, I'd like to move on to the phase after widespread vaccination and community infection. Of course, we had by November 2021, there had been 248 million COVID cases worldwide, and 3.9 billion persons had received at least one vaccine dose. And Omicron was identified in Africa in November 2021 and became, by 2022, the most dominant variant of concern. So this was a variant of concern designated by WHO. So, and Omicron actually continued to evolve subvariants even up to the present day. At the present time, or until very recently, Omicron BQ1 and XBB were the most rapidly spreading strains. And the feature of Omicron is that the spike protein contained multiple mutations which were responsible for evading antibodies produced as a result of vaccination with the spike protein, as well as the monoclonal antibodies used to treat clinically ill patients in hospitals. So the important point to remember is that COVID-19, of course, infection elicits antibodies against other antigens besides spike, whereas the mRNA and then the AstraZeneca adenovirus vaccine be based on S, so there's all S protein vaccines elicit only antibodies against the S protein. This is not the case with inactivated whole virus vaccines. So at this time, the driving forces for selecting variants, especially Omicron variants, was the evasion of adaptive immunity, that is the antigen-specific immunity, particularly antibodies, and increased transmissibility possibly, although this has not been clearly demonstrated for Omicron. Next slide, please. The IDFISH and IDGenics are involved in primarily tick-borne diseases, the diagnosis and of tick-borne disease, but also during the pandemic, we worked on the immune response to COVID-19. And this slide is illustrative. Here, these are sera from patients, and we're looking at sera. We would love to look at the upper respiratory tract mucosa, but that's very difficult. Some people have done it. And we looked at IgM antibodies and IgG antibodies against the S1 domain, which includes the RBD, the S2 domain, the isolated RBD, and we looked at IgM antibody formation, IgG antibody formation in cohorts of patients at varying time intervals after they had become PCR positive. And I'd like to just draw your attention to two features. One is the IgM antibodies. The black lines show the proportion of sera that were IgM positive. That decreased and disappeared in our hands when we looked at sera over 100 days after diagnosis by PCR, after infection, in other words. The IgG antibodies reached almost 100% prevalence by days 50, in between days 51 and 65, and continued to remain high after 100 days. The other feature I would like to draw your attention to is that there was significant response to the nucleocapsid protein, which is the N protein, which you don't see when you have a spike-only vaccine. And that was quite strongly immunogenic. The RBD alone was not strongly antigenic. In other words, it didn't. Next slide, please. I mean, we've gone back a bit. Now, can we go back to that particular slide I was talking about? That's it. Thank you. So the RBD itself didn't appear to be terribly strongly immunogenic, but that may reflect the fact that we were only using a small part of the RBD, and the S1 would also contain the RBD. So this is just to illustrate some of the aspects of immune antibody response in infection, after infection, and people had COVID-19. Next slide, please. We'll skip this slide. Next slide, please. Right, thank you. Now I'd like to talk about the Omicron BA.1 variant, which arose quite early and was very widespread. And there were some very interesting studies that were done on this. It has a high affinity for ACE2 compared to the previous variants, evades therapeutic monoclonal antibodies and vaccine-elicited plasma antibodies, not completely, but partially. Surprisingly, it had lower replication rates in the lung and gut cells in vitro, but similar rates in nasal epithelial cell cultures compared with Delta. In other words, it seems to prefer to replicate in nasal epithelium. It also induced a lot more type 3 interferons and interferon-stimulated genes, particularly in lung cells, compared to other variants of concern. So this would indicate that it wouldn't be terribly pathogenic in the lungs. And it also used TMPRS2, the transmembrane protease, for cleavage less and used the endocytic pathway more. So that led to decreased syncytium formation in the respiratory cells, and this would also contribute to lower pathogenicity. And some elegant work has shown that the non-structural protein 6, which forms the endoplasmic reticulum membrane vesicles where the virus replicates, the RNA is replicated, and which is and NSP6 also has a role in pyroptosis, or the death of infected cells. And these effects of NSP6, mutations in NSP6, leads to reduced pathogenicity, which is very interesting. So BA.1 was less pathogenic than Delta. Next slide, please. although it was more fit. It was able to spread more rapidly and evade antibodies. So at the late stage, when there was widespread community infection and vaccination, the driving force for selecting variants appeared to be the ability to evade antibodies in the population. However, many of these studies use virus neutralization assays to measure the effects of antibodies on the different variants. And the virus neutralization assay really relies on blocking the spike ACE2 interaction. But antibodies can act in a number of different ways besides blocking the spike ACE2 interaction. It can act via complement-mediated lysis of the virions in infected cells, natural killer cell-mediated killing of infected cells, and phagocytosis of virions in infected cells. And also we have T cells, helper T cells and cytotoxic cells. T cells are able to bring about protective immune effector mechanisms against infected cells. So these are not assayed for when you do a live virus neutralization assay. An escape from T helper cell immunity is much more difficult than escaping antibody-mediated immunity, because we have a huge repertoire of class 1 and class 2 HLA antigen-presenting molecules, which can present different T cell epitopes. However, what has been shown is that after spike-based mRNA vaccination, high neutralizing antibody responses to spike and abundant effector memory T cells, T cytotoxic cells recognizing S, are correlated with better protection after S-based mRNA vaccination. So, and we are also fortunate that there's no evidence yet to show that Omicron significantly evades T cell recognition of spike protein. Next slide, please. I thought I'd show this rather nice work, which illustrates the importance of, which presents some of the data with live virus neutralization in different variants of very different variant viruses. So these are this assay measures antibodies that can reduce the development of the virus in an in vitro assay, using different dilutions of the antibodies of sera obtained from persons. So after 3 doses of mRNA vaccine, the sera of these vaccinees seems to neutralize the ancestral strain at a higher dilution, 2.5. So the higher the theta, the better the antibodies are, compared to the more recent Omicron variant. So much better than, say, XBB, which the neutralization theta is just barely above control. This becomes better after 4 doses of an mRNA vaccine based on spike, but not very much in the case of BQ1 and XBB. But interestingly, after 3 doses of mRNA vaccine plus a BA2 infection, the sera of these people have a higher, significantly higher neutralization titer against the ancestral strain, but also better neutralization titers against the progressively recent Omicron variant. So the BA2 is older than BA5, and which is in turn older than BQ1 and XBB. So this, I think, shows the ability of the Omicron variants that have risen progressively to progressively escape antibodies produced against spike through messenger RNA, spike-based mRNA vaccines, or even an Omicron infection. And actually, a single Omicron infection seems better than a 4th dose of an mRNA vaccine in people who received 3 doses of mRNA. So this, I think, illustrates the escape of Omicron in the more recent variants, progressively greater escape from anti-spike antibodies, which is due to mutations in the S protein. Next slide, please. Now, how do we address this? One approach that's been used recently and the data published from Nordic countries is that instead of using the S protein from the ancestral strain, one has the fourth immunization in these countries used a bivalent mRNA vaccine, where either BA1, Omicron S, or BA4, BA5, the same S, the same sequence, spike protein sequence, with the ancestral strain. So it's a bivalent combining one of the Omicrons with one of the ancestral strain, with the ancestral strain S protein. And these were introduced in 2022 to try and overcome the shortcoming that we saw earlier, which is reduced viral neutralization. And this led to significantly better protection against hospitalization and death from COVID-19 in Nordic countries. However, we must remember that vulnerable people, particularly the elderly, will remain more susceptible to severe infection and death despite vaccination and the reduction in pathogenicity of the presently emerging Omicron variants that we're seeing. Next slide, please. Finally, I'd like to close with some perceived research needs. I'd like to change this a little bit and say that we need, go on to this point, that we need new approaches that can very rapidly generate clinically useful antibodies for emerging variants of concern. Should the Omicron become even more, evolve more variants or variants arise by other means. And such methods where you can, instead of using producing monoclonal antibodies, you can do some synthetic work. You can either go for chimeric antibodies, antibodies that have able to bind two different epitopes on the S protein or other proteins, nanobodies, which can be generated much faster, made up of single variable regions of antibodies, or even broadly neutralizing antibodies. And finally, I'd like to I'd like to mention one other point, and that is that we need to continue to monitor the immunogenicity and antigenicity of emerging variants of concern and optimize COVID-19 vaccines appropriately. And finally, we need to be aware that SARS-2 has adjusted to develop in wild deer populations in North America recently. And of course, there are related viruses circulating bats and mammals in Southeast Asia. And if you have co-infections with a human SARS-CoV-2, then the possibility of generating genetic recombinants with increased virulence to humans becomes a possibility, real possibility. And this is what happened with swine flu influenza in 2009. We had recombinants generated in pigs. So continuing monitoring of the sequences of SARS-2 variants and their properties is important for maintaining vigilance against this virus. So, although the pandemic is now officially considered to have been over. Thank you very much. Happy to take questions.